wabarakatuh.
Waalaikumsalam
warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Alhamdulillahirabbil alamin. Wassalatu wassalamu ala asrofil anbiya'i wal mursalin Sayyidina wa maulana Muhammadin wa ala alihi wa sahbihi ajmain amma ba'd. The person in charge of the Parasite Protozoology course, Mrs. Novita Eka Putri SSIM Biomed, we respect today's webinar speaker, Dr. Meherunisa, PhD, and to all happy webinar participants. We respect today. Praise and gratitude let you say to Allah Subhanahu wa Ta'ala, has bestowed and His blessing and gift, so that we are still given the grace to be able to meet in expert lecture activities with them comparison and evaluation of various methods of malaria diagnosis and assessment of hematological parameters in malaria infection. Prayers and greetings we say to our Lord Prophet Muhammad Sallallahu Alaihi Wasallam, to his family and friends, may his mercy reach us until the end of time. Furthermore, let me Widyawati B. Anuna, as MC, will deliver the schedule of activities today. Opening, singing the national anthem Indonesia Raya and Mars UNISA, remarks by the person in charge of the Parasite Protozoology course, presentation of material and discussion, and closing. Well, the first event is the opening. Let us open and explore lecture activities with the them comparison and evaluation of various methods of malaria diagnosis and assessment of hematological parameter in malaria infection by both saying Basmalah. Bismillahirrohmanirrohim. Next sang the national anthem Indonesia Raya.
Wahai seluruh warga UNISA Yogyakarta, bulatkan semangat baktimu. Siapkan generasi unggul dalam prestasi, berjiwa profesional berani. Iman ilmu kuatkan, amal mulia kerjakan, akhlakul karimahmu berkemajuan capai cita. Itulah misi kita bersama sebagai civitas utama. UNISA Yogyakarta, raih citra sempurna, sentosa abadi dan jaya. Ridho Tuhan selalu ku minta, anugerah limpahkan selamanya. Ridho Tuhan selalu ku minta, anugerah limpahkan selamanya.
Next is a speech by the person in charge of the Parasite Protozoology course, as well as who will open the activities today to Mrs. Novita Eka Putri SSIM Biomed is welcome.
Assalamualaikum warahmatullahi wabarakatuh. My respected Dr. Mencerdaskan, PhD, my respected Institute Aulia Muawana, SSNSD, Chair of the Medical Laboratory Technology Study Program, Asia University, and the all participants in today. First of all, we would like to thank Allah because we were able to attend the event in this morning. It is great pleasure for me to welcome you to expert lecture with Dr. Mehru from Kuala Lumpur University. I am very thankful to you, Dr. Mehru, for taking time out of your busy schedule to attend this event. I hope the participants can enjoy and learn from today's expert lecture. We can learn from Dr. Mehru about protozoa examination, especially about malaria and assessment of hematological parameters. I would also like to thank the organizing team who helped organize the expert lecture today. Let's enjoy today's expert lecture and thank you all for being a part of this lecture for us, and I wish you all a happy learning experience. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.
Next is the delivery of material will be led by the moderator, who serves as moderator is Hasna Hasnifa. To the moderator is welcome.
Thank you to the MC for the time and this that has been given. Let me introduce myself. My name is Hasna Fiza Azara as the moderator at the Protozoology Parasite Expert Lecture this morning. At the Protozoological Parasite Expert Lecture, the title, Compassion and Evaluation of Various Malaria Diagnosis Methods and Assessment of Hematological Parameter in Malaria Infection, with Dr. Mehrunisa, PhD from Kuala Lumpur University of Malaysia. Before entering the main event this morning, let me, as the moderator, say a little sum of the CV of our presenter this morning. Academic qualification, PhD in Medical and Healthy Science, International Medical University, at 2015, MSc in Medical Microbiology, University Science Malaysia, 2010, BSc in Biotechnology House, IMST University, 2007. Professional membership, Malaysia Society of Parasitology and Tropical Medicines, Assistant Secretary, 2020. Women in Science for the Developing World (OECD) adopted counseling member 2020. Malaysia India Education Development Association Fiance Auditor 200,000 2020. Technique Teaching Activists Page Methodology Materi in Applied Science Medical Parasitology Etymology Basic Microbiology Environmental Microbiology Scientific Communication Endangered Parasitology Basic Microbiology for Environment Health Diploma. Maybe that's all I can say about his CV, which of course has many more achievements to short the time, and because the presenter this morning is here, let's get straight into the main event, mainly the protozoology parasite expert lecture on comparison and evaluation various methods of malaria diagnosis and assessment of hematology parameter in malaria infection with Dr. Mehrunisa PhD. Time and place we in video.
Good morning. Can I start?
Yes, Doctor.
Oh, thank you so much. Okay, a very good morning to the organizing chair, to the moderator, and also Dr. Nukita Putrina. Thanks again, once again, for inviting me. I think every year this is my fourth time since
Yes.
COVID. Thank you for your consideration, and it's my pleasure to meet all the young students of UNISA, young, energetic. And I believe some of you have come for the first time. So the topic was given to me on malaria. I'll try my best. Okay, I'm a parasitologist and produced by the data and the chat. But my main research is from helminthology. But I'll try my best to deliver lecture. Okay. So are you all students here? Just check. Is that all students?
Yes, Doctor. This is student of from University Asia.
So if students means, can I be more to like student type lecture
Yes.
rather than a conference talker? So I will be like student type, and I will speak a bit of Indonesian language. Okay. So let me share my knowledge. Okay, so if student, I can be a bit informal. So I believe you can see my lecture notes. Yeah. So, Daria. Yeah, very good morning. So today our talk is on comparison and evaluation of various malaria methods. So before that, I would like to ask permission actually off my video because it is dragging my and the video was getting slower on my side. Then we will move back. So selamat pagi once again. So apakah dia malaria? I believe some of you have learned in your class the basic thing. So what I will do is I will go through the basic knowledge about malaria, and I will focus on the diagnostic method. Okay. So you know malaria is a mosquito-borne disease. Penyakit bawaan nyamuk. Kalau kita lihat di sini, ini merupakan nyamuk Anopheles causing malaria disease. Untuk dengue pula is nyamuk dengue. So today my outline for this presentation will be on various methods. Semua method kita akan lihat dan juga on hematology. In the blood slide, what can you see? Different species of malaria. Okay. Is my voice okay?
Oke, dokter.
Thank you. Thank you for the information. So, it is. Okay, let us look at the history of malaria. Malaria parasite adalah dalam kategori sporozoa. Spores. They don't come like helminths, egg, or cyst. They are spores. And they are transmitted by Anopheles. So the mosquito yang anda lihat now ada perutnya warna merah. Okay. Malaria disease is only caused by female Anopheles, pregnant Anopheles. Kalau nyamuk jantan dia tidak menyebabkan penyakit. Hanya the female mosquito when they are hungry, they need to take blood. So bila dia ada penyakit malaria dalam bentuk sporozoid dalam tubuh kita, bila dia menggigit kita, penyakit dia akan. If we lihat cara penyakit malaria parasite was founded by Charles Laboratory. Dan wajah mereka, dia actually a branch of the soldiers of policy. Then they are right. Malaria parasite, the mosquito, mainly lives near the forest. Those army soldiers atau pihak-pihak yang bekerja dekat hutan, they have more prevalence to get, high chances to get. Next, staging dia. Macam mana kita stage cell stage, Leishmania stage was developed by scientists Wesley, Italian scientist Pico. So hingga kini, we have 4 species malaria, falciparum, vivax, malaria, open. Satu lagi yang tidak ada dalam in all the 4 species, kita ada falciparum and vivax yang menyebabkan 95% of infection. So age distribution, ini merupakan satu yang data saya dapat dari publication terkini di Kalimantan Indonesia region. Malaria distribution, tapi yang ini tidak ikut species dia, just the general malaria distribution. It's on 2019 and 2020. We see umur yang paling banyak dapat malaria is 20 dan lingkungan 20 and sebab kalau kita tengok ini merupakan umur yang mereka bekerja di luar, actively bekerja di luar. So, mungkin sakit-sakit ini dia kerja di kawasan pedalaman atau hampiran dengan hutan, so they got the malaria infection. Kalau kita lihat umur yang lebih tua ni kuranglah pendedahan mereka kepada penyakit kurang. It's actively working. This is mosquito here. So they have a proboscis, abdomen, head, thorax. and- So as I said, we have 5 different species. If students, you look at the CDC website, you only can see falciparum kat WHO World Health Organization website. Hanya ada falciparum, vivax, overlay dan malaria. Kelima ini is knowledge. Knowledge is very dangerous, but it's caused by apa? Monyet. So that's why we don't, the WHO don't consider them as a human disebabkan it, but this is dangerous. In Malaysia, this is quite high. If we look, falciparum adalah paling berbahaya. Severe harassment. Vivax worldwide, overlay, you can get into the picture. Malaria is a tropic. Seorang pesakit yang pernah dapat apa-apa malaria, they can get sorry. So let's see the description about each malaria species. Vivax and overlay. Now, these are the parasite species which can go and hide dormant in the liver. Dia can hide in the hati and dia boleh jadi aktif lepas berapa bulan. Malaria pula, they can cause long-lasting infection. And lifelong, the patient can get symptoms. Malaria and non-inside. Parasite. Can I proceed? Is my audio okay, students?
Yes, Miss.
Sorry, sorry, Dokter.
No, no. And malaria and non-inside, it's like dia nampak hampir nampak have really big. Dalam semua species ini, yang paling cause complication is plasmodium falciparum. So plasmodium, it has 2 hosts. 2 rumah. Satu nyamuk. Bila kita tengok kita hidup malaria, the parasite actually develop the animals. Di human pula, setengah life cycle will be developed here and completed by the. That is the reason. One of the best way to reduce malaria is reduce the get rid of the tapi reality nak membahas speed yang bukan senang, especially anomalous or disease. So can do prevention like fogging and so on, but not give. And it's mentioned that as we look, if you look at the life cycle, the sexual reproduction actually takes place in the mosquito. And parasite is transmitted to human when we take the food needed to develop the malaria. So before we go further, there are few kata kunci yang kita kena ketahui untuk pengetahuan malaria. We have the word like incubation period, fever pattern, relapse, recrudescences, and infection. These are all the important terms yang anda kena mengetahui pelajari. Term ini is very important. Incubation period, so following the bite of the infected mosquito in the first okay, let's say today myself, hari ini saya pergi dekat perkampungan nasi, nyamuk Anopheles, which is carrying Anopheles, malaria parasite bites me, they get bitten. Hari kelima, saya ada demam. So incubation period for me is the infected mosquito, the following the bite of infected mosquito. Sebab selepas hari kelima, baru I dapat. So this is called incubation. Fever pattern is length of time needed for schisms to burst in one side. Schism is one of the stage. Berapa lama dia akan pecah? So this is important. Sebab when schisms pecah bermaksud for RBC pecah. When RBC pecah, patient get fever. So that's where your genetic RBC red cell. So that we call it. Then you have the word recrudescences. Recrudescences is parasite reappear in the circulation after low detection. Bermaksud, okay, let's say you pergi, patient dah discharge. Patient dah discharge because of the blood split there, no more malaria parasite. After blood split, maybe one time, two times, or okay. So patient can be discharged negative malaria. That is very low. And suddenly, so the patient go back again, fall sick, high fever, must hospital, tengok stuff. So this is actually can happen why back. They actually high to normal. So the more of time to be and it back. Then we have the term reinfection. I'm cleared from malaria, I go back to my kampung and another Anopheles not advised. So there is reinfection. Because my area has relapse. Relapse, this is again with Vivax and Overday. dalam bentuk hypnozoids, the parasite itu dormant dalam peti liver, suddenly it appears. These are few terms that the student will be studying. Not you get confused because the importance of the terms here is every species of malaria have different incubation. Even that's why it is the so fever pattern. What is the fever pattern? Then doctor needed is on the one. All the fever pattern here. Palsy 7 to 12 days. But patient here. Fever pattern berapa? 3 to 36 to 48 hours. 1.5 day to relapse. What is relapse? We call back. Relapse only happens in Vivex and Vivex blood. Fever pattern 48. Incubation 10 days to 6. Sebab mereka boleh memerlukan diri. Relapse. Malaria it's 72 hours. Fever pattern boleh 48 hours. Monkey malaria it's very dangerous because every 24 hours you dapat pecahan. That is why we say relapse. Relapse hanya for 5 days. Okay. Mereka boleh morok di looking at incubation period. So falciparum quite fast. Symptoms 7 days, followed by northern sign. Then this is why I say you need the important terms. So I believe maybe some of you would have learned this. Maybe just yeah, that's the picture. So the story starts with this. Malaria life cycle complex. Tapi menarik. So, let's start from here. The ekor, mosquito, Anopheles butina, yang ada yang pregnant. Usually pregnant, they need hemoglobin, so they bite human. The male mosquito, they just take some plant ribs, that's all. They eat. So when the female mosquito bites human, they release what? Sporozoid. The this is the infected mosquito malaria. So the first injection yang dia akan bagi pada manusia dah so sporozoid will directly go where? Liver cells. When you inject, first target. So knock, 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 liver cells, are you there? When it's trapped, the liver cells. So they develop into schizons, they rupture the liver cells, ruptured schizon. So once they rupture the liver cell, they akan release virus. So yang ni belum masuk dalam darah lagi ya, akan dikerja. Then dia pergi to human blood stage dalam bentuk schizon. So once go here, call that as virozoid. So these virozoids will go to a second stage called invention proposal. Yang ni yang kita boleh tengok banyak di dalam the big stage. They develop, then they develop into schizoids. And this is high in schizoids. They rupture and release the protons. This is asexual. For those sexual cycle, they will put mutual proposal ni akan jadi lebih besar and develop into chemicocytes. You can see clearly dua pathway lah. One is from the asexual cycle, one rupture. After rupture, it's only like your temperature and move up and down. Chemicocyte ni boleh jadi female and female. So the type manusia, they will stop here. Chemicocytes. Then mosquito anaphylaxis bites the chemicocyte ni, dia akan fuse badan anaphylaxis mosquito occipits. Occipits will develop further into seeds. It is protozoid kita yang ada dalam peraliran darah, di air liur, anaphylaxis mosquito. So that is why when anaphylaxis mosquito bites you, they release spores. Okay. So it's clear? Have the exoepicycle. You have the human cycles. You have the cycle. You see clearly. In human, they only can stop until it is. The fusion can only, development of the sporocyte can only be done by the. So that's why when we do the blood test, if in the clinic, hospital, we manage to detect chemicocyte in the patient, immediately you need to inform the why? If a patient has chemicocyte, they can spread to another patient. And the mosquito bite chemicocyte will form. So we know that if the mosquito bites the patient that under the stage, we will develop a that is the reason immediately for agents, other prevention. So some of the clinical symptoms of malaria, you have cerebral malaria, pulmonary, acute renal failure, severe malaria, bleeding. On the clinical implications of that, I won't be happy so much. The overall I'm sure if any one of you has malaria before, but malaria is very dangerous. They can cause miscarriage and death. So now we have medication. But has to be given, but only help people. And about vaccine, the research is still ongoing. They say yes, clinical trials is very difficult to develop a vaccine. The reason is malaria has been technological on. So before I go into deeper into that, I'm sure you have or interviewed the belief that blood cells like fat matter and we will all have practical examples. Till now, the belief blood cells one of the most reliable methods to type golden standard skin microscope. When we do the hematological analysis parameters, white blood cell patient will not have drug, but you won't know if the patient has them until you do it. So we still rely on the blood cell. That is very easy. It's microscope. So as we know how it is like, we have to. So when we want to understand that malaria parasite, so we have a big stage. Protozoite, these on mentioned when neurons rupture, will get you will develop rupture because inside neuron are the neurozoid. Neurozoid is mirror. So yeah, species area. This is when the rupture keys on. So how you identify the keys on stage, you can actually count the neurons. Then gametes. Gametocytes are so this is the actual formula. What is not showing you the figurative form. This is actual form in the smear how we form trophozoites. The trophozoites are quite big. Gametocytes can have either banana shape. This is can calculate. So this is the overall picture for malaria, and each species will have different morphology. So that is challenging. You as a lab scientist technician, easily you can report a patient in malaria under the country. But the taboo is the species Melissa and malaria virus. Okay, you need proper screening. Now, so let's start. First is Fancy pada di malaria itu. Whenever you look at the smear, always compare dengan darah dan right. So first, infected malaria, non-infected cells, hide from it. Dia akan ada titik the infected cell calling moral. It can be to find and to proceed with it, but not so much. And Propozoid compact Kizon. Kizon, the six from Proton, the non-self. So, particular, first thing is affected activity, save life. The propozoid, dia nampak very uniform. First is the easiest in all the phases. Why is that? Why is that? The infected are compared to non-infected. Compared dengan particular healthy article here find dot 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 dot. Okay. And it has the people. They have that and keep on around 12 time. So the moment you see the infected RBC and touch, so your mind is first the Wi-Fi or why overlay? Overlay same like infected RBC can much, but not so big, not so big. What we call that? Dot, 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 yeah. Okay. Here pula, the RBC, sorry, the season inside the seasons, we also we can complete chemical cycle. You can see a propozoid that had a it's like this from the influence. So last but not least, malaria. Malaria. Mana sebut malaria, maka dia akan target darah old blood. So I think human darah muda itu lebih banyak and young blood produced. The old blood level. So this malaria only target old blood. That is the reason. Bilangan dia akan always very high. Kecut. Affected one even normal decrease. Pasti pada same time affected virus and overlay and touched and P malaria. dots, we call it as the cipher. So I have also put the table to differentiate each of the malaria species. Can you see? Right? Vivac over there and that infected RBC right? Infected RBC. Multicolor black. Vivac brown distorted. Overlay inflated etc. Simply effect RBC. So we call for some amount of fat, separate dots, big spot, each of dots. Propozoid or big, right? Chromatic dots. It's not so important. Nature schizon. This is the good indicator in hematological parasite. Nature schizon. You can count. The moment you count, you can differentiate X, hundred, and gametocyte, particular that went to a table like this. Easy hematology. So this is the male picture. It Vivac over the falciparum ascending. Propozoid. How it is. And one more thing about the malaria. The malaria, if you really look, dia akan a tinge of, dia akan ubah sikit brownish to yellow. Propozoid. And dia ada macam basket shape, bird shape or this. It, the other way, it's not can be good. Easier to type of particular and followed by cross violence. So sometimes students sometimes confuse with the propozoid and the minozoid folic acid here. They don't be confused with propozoid. So the only one way for you to understand other gametocyte, you will be filled, all the inside part of the parasite will be filled. That is one. So are you all okay so far? Students? Shall we take a 5 minutes break or I continue deeper into all the laboratory diagnostic malaria? 10%, are you okay? Shall we take 5? Dr. Lupita, shall we take a 5 minutes break? Okay, just take 5, right? Thank you. So we'll just take a break for minutes, 5 minutes, then we'll continue. So we will see at you for my time 10:58. So just drink water, go to a washroom, and please come back, students.
Untuk teman-teman semua, mohon ditunggu sebentar ya. Kemungkinan ada masalah sama jaringan pemateri.
Okay, can you all hear me? Yeah, we shall continue.
Oh, Dr. Mehru.
Yeah.
Yeah.
So we will continue.
Yes.
Okay, now the second part we will look at the current laboratory diagnostic that is used. So we have seen the hematological parameters, how each species is type. Now we will deeper in right various methods. As I told before, the golden standard technique is still parasite. We are still technique yang paling utama berbanding dengan teknik. So, we will look around at 6 techniques. Kita akan tengok lebih terperinci diagnostic, followed by quantitative barcode, immunography, then rapid diagnostic kit, enzyme linked immunosorbent assay, flow cytometry, It's all one by one. First of that, microscopy, what we need to do the blood flow. Blood flow, thin blood smear and thick blood smear. Untuk pengiraan parasite, we use thick blood smear. Pengiraan thick blood smear bermaksud just put one drop without thick blood smear. Actually, the RBC akan rise lah. We only depend on the WBC. And thin blood smear, kita gunakan, thin blood smear yang we tarik until we use to diagnose various species. Thin blood smear untuk detection of the species. Thick blood smear is untuk so, how do you calculate parasite? So, the method that I use is I refer on darah lymphocyte and we count based on reference because in thin blood smear, RBC you can't see. The RBC macam ghost cell, Vivax boleh saat the first and foremost need to examine the 100 medication and area. Right. If not malaria parasite, see the virus, right? Then move until 200 microscopic. Langsung tak ada, then kita report negative. It will be reported as malaria parasite in 100 weeks. So when kita melihat pada slide, ya, you have to calculate everything. Parasite, you need to include everything. Also white blood cells. What you can do, can actually divide them into one. So this is the whole criteria that we calculate. So just read it first. First and foremost, scan the whole view of the slide. Now let's say this is the slide. You need to scan the whole view. Now, don't just scan the center of the view. Scan before counting. Hari ni you dapat slide, jangan straight ambil handphone. No. You need to look at the overall. Divide the field into quadrants. Okay. Only count WBC, not the stages. Okay. So this is the differential blood counter. Okay. So, I'm not sure if you're using this in your lab. We use this. So each time for white blood cell, we click once. So malaria parasite, we click one. Gametocyte, we click separately because you have to key in the number of gametocytes separately. So this is the differential blood counter. You can use the hematological blood counter for that. So how we do malaria vaccination? So I'm using, I was trained, refer to white blood cells. The number of leukocytes is used is 8,000, assuming an average normal total leukocyte 8,000. So my reference is I use leukocyte cells, and we use 8,000 cells because the average is. So how do we do? First and foremost, you calculate the parasite until 100. Let's say you tengah calculate like the super parasite. Make sure you calculate 200 white cells at the same time parasite cells. Kalau parasite yang dijumpa dah melebihi 100 atau sama dengan 100 dalam 200 sel detik, stop. And record the results as per 200 parasite. 200, how many parasite in 200 white cells? Right. The way, I hope you are not understanding here. In 200 white cells. Then you use the. Kalau anda dah sampai 200, even 200 white cells dah sampai, tapi parasite ni belum sampai lagi. Dia just 40. In 200 white cells, what you do? You continue counting sampai 500 white cells and record the results. Now, if you have counted less than 99 parasite, 500, stop counting the record the sum as 500. So this is the parasite count for white blood cell. White blood cell count can be either 200 or if I see the thing 500 here, I know the patient has very less parasite. That is why the scientists went and count until 500 white blood cells. If here I'm looking 200, bermaksud patient ada banyak parasite. Sebab tu dalam 200 dia dah stop dah. That is the indicator. Let's try this. Let's say you get a patient, asexual stage dia 120, sexual stage 40, sexual stage medium stage dan side. How do you calculate? How do you? So first, as a scientist, what you should do is you calculate the asexual stage. What you have to do, you take 120, then you calculate them. So asexual stage, what it can be? Major trophozoite, schizo. All these are major asexual stage. Divide by 200. That means, sebab you see, the asexual lebih daripada 100. So that's why they stop the calculation at 200. Darah 8. Next, determination of sexual stage, the same formula, just replace it as 4. Divide by 200 and times 8,000. Assuming 1 will be 50,000. So how you report? So you report in the form, the patient has 4,800 asexual over 160 sexual cycle per microliter. So not microliter. But this the number you need to have. Clinicians, they need to know the burden of parasitemia, malaria in your health. If they don't know this accurately, the dosage of treatment to the patient will be second method. So far, I hope you are clear about the diagnosis. Second method is quantitative buffing. We call it as QBC. This is something like malaria, but sorry, a microscope. See, as what you see, a microscope, as I say, is the golden standard method. The only thing is the patient interpreting the slide is the other malaria problems. Malaria scopies is not a easy job. Job that need patients perseverance and a high skilled knowledge to read the slide. Quantitative buffing, fluorescent microscope of the acrid orange coated capillary means by so the difference in technique between this, it is still used. You need to use a capillary. They have something coated called A2, arcadian orange, and they are a bit more sensitive than that. But they say 55 milligrams. How you do? You can either take the patient's urine, venous fluid, they put and they spin. When you spin, they will be separated platelets, side, sides, and the sides stage separate. Here. We call it the QBC. If you see, kalau untuk light microscopy, have to do the blood smear yourself, then that slide you kena tengok semua cells kan. When you do the blood smear, you will see the white blood cells, you will see artifacts, you will see what else you will see? Something and so on. Yes, so this is how the WBC. But when you use this when you use this QBC, the capillary itself will be separating and helping you separate different layers. So what you can avoid, just masukkan dalam fluorescent microscope like this and directly see the tube. Just this particular area you focus the microscope and then the slides. So that is the benefit of the using QBC method. Any questions so far? This is up to and so the it's the source. So this is very nice. Just you have to speed and gently. Looking at the benefit, Memang be compared to microscopy, it is very easy because you can take large big number of samples, just fill the capillary and speed them. Quick and save time. Very good. Requires centrifuge, special state. Because it needs centrifuge. Okay. What are the disadvantages? First and foremost, this one can detect the parasite here, but to really look into the species, macam species, because it's the fluorescent, it will die. Second, obviously the capillaries are important. These capillaries are good in QA. They are not cheap, they are expensive. Like, of course, compared to your microscope slides, how much it will cost. It's a box around 8 dollars for the Howard slip, of course. Compared to this sophisticated strip, sophisticated strip, so will it be feasible for underdeveloped countries having high prevalence? Can they buy this capital? Not that question. The capillaries can't store the data preferences. Capillary can't be stored for data preferences. See, kalau slides, kita boleh simpan untuk beberapa tahun. Actually 1 year lah, 1 year, 1.5 year. It's very good. You can keep as reference, but this, because this is actually a capillary, you can't keep it. And because you are not putting any fixing it, the RTD akan muncul. Okay. Third, we look at immunotherapy-based diagnostic tests, RTDs. So, this RTDs has been very famous since masa COVID. RTDs are rapid diagnostic test kit. Those days, RTDs, only doctors can use, scientists can use that. As masa era COVID start, you can just buy the test kit, test, and cook yourself. That is how RTDs have been so famous. But we don't have RTDs sell to public malaria. Because malaria, you need like COVID, just take your nasal swab, throat swab. It's different. The principle is RTD enzyme protein. And it's immunosuppressive and cytotoxic. So all these are the immunochromatographic based tests. So this is how we decide. And so you have so what happens? It is said in the last decade, RTD was used as an alternative diagnostic method for malaria. When it happens, when a good microscopic quality can't be provided to one. So this always happens when we have lack of good microscopics. With malaria microscopy, not easy, and you always need to refer to someone. The best you need someone to guide you your eyes. can be very confusing, it can be tiring, sitting in the chair for a long time, and you can have back pain, ergonomic issues. Right. So when they found RTD, everyone got excited because RTD is based on the presence of antigen and easy. This RTD is a natural flow immunochromatography test on the nitrocellular strip, which detect the antigen escapes. So, a patient infected with falciparum, we have falciparum antigen, others with this like Vivax, malaria, overlay. So, this is how it works. The RTDs in general, not only for malaria, even for all the other hepatitis, you have a nitrocellulose membrane at the base. Then you have the conjugated antibody. In case of malaria, they put some kind of proteins related to malaria. Analytical, where you drop your sample, they have an analytical here. And by capillary flow diagram, you can imagine all this clearly. All of us have been malaria, COVID tested a few times. Okay. So this is the principle by the yes. They have test lines. If you look for malaria, some kits they come with 2 test lines. Control line is to validate the QC, say whether your test is successful. Okay? Test line for malaria will be 2. So if you ask me why, you should. If you're using one malaria and falciparum kit, that means kit B boleh specifically detect falciparum and other malaria. Tapi untuk protein falciparum hanya satu line, protein line. So if the patient is positive 2-2, 3-3, bermaksud the patient has falciparum. They're just positive control line, then could be the patient positive. Therefore, that is the right. You can read here the explanation. And also it is said because we use 2 different immunoglobulin, IgG and IgN for malaria and antibody. So RTDs are very rapid and helps us a lot. Okay. You can see here. This is example of RTD vaccine. At times, some kids require you to put a buffer. Buffer ni actually menolakkan your sample from blood. Sometimes the blood is very thick. Again, patients is very thin. So you need the buffer is actually thick. Sample is the process. Drop by the free blood have anti-falciparum antibody, anti-malaria. But nowadays kids they can be specific for malaria, but other antibody is bigger. So that's why we put everything in. So if it is negative here, positive here, it means it can be positive. You can see this is example of negative. Because why negative? Only the control line is lighted. In non-falciparum, you have the control line and anti-malaria antibody highlighted. Is a pure or mixed breed infection, you have the patient here has all the antibodies within the two. For macam ni, very test because the control line did not light up. So in the case, you may ask, why doctor there's an invalid happening in that diagnostic? Well, it could be a few reasons. First, check the expiry date. Make sure that even expired but always for diagnostic. Right? If you just want to practice, other than that, my recommendation is that's not for diagnostic for six patients' life. Invalid can also be caused by your technical error. How much of blood did you load? You just load little bit, or the blood itself has some chemicals infinitely mixed that you cannot run. Or it could be when you are doing tests, you keep on shaking it, or the table is not stable. Somehow the buffer is not adequate, expired buffer. Various reasons can cause that. So how do we overcome this? So for invalid tests, we just repeat back the test using. Don't repeat immediately. Just wait for 1 hour. You can open a fresh test kit, correct everything, start fresh. That's how it covers. So, what are the common protein targets for malaria arthritis? Malaria arthritis, these are the proteins that are captured within it. So, you have Plasmodium falciparum histidine rich protein 2, histidine falciparum lactate dehydrogenase. All these are written in 5X specific human falciparum LHD, it's what proteins used. Enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay, like the UNISA, has been developed as a validated method to detect BH2. What is this? It is in blood plasma envelope. It's so one is the previous was actually this one RTDs. RTDs, the same protein kita boleh untuk for ELISA, we target 2 immunoglobulin. Antibody immunoglobulin apa? IgG and merozoite specific IgM. Plays an important role in response to parasite at the stage of immunity. So malaria patients, it is found that they secrete IgG and IgM, and they have specific the viral specific IgM. So bila kita buat ELISA, we will target against these two antibodies. So how is it done? Same method. The difference is here there is no kit. You have ELISA antibodies. The kit will come with the ELISA kit. Immunomodified plate for degree Celsius. First, you lyse the blood and you mix the blood. First. And second, with malaria antibody, incubate and you detect using a microplate read, the common plate reader that you use for the MDT any ELISA based. So this is just one of the example. Various lab settings might run it, which target different, the protocol might change here. But should be based on IgG, the next is flow cytometry. Now, if you see, they are going from the manual method slowly the high end method. Flow cyto is automated method like malaria by scientists for 1 year. The blood sample are mixed with the spirit content formaldehyde because the RT dyes and white blood cells. Flow cyto is this is one of a high-end method of flow cyto. And here boleh detect exam from I did flow cyto. We don't have the machine here, but I went with another experience that that popped up in that. But flow cyto need everyone can do it, but to understand the principle of flow cyto, and he or she should have been well trained because the data is quite a lot, and also you must do a proper flushing. The dye is not like easily must and the chemicals are being parasites and white blood cells are distinguished counted based on two column fluorescent and its words are actually used to the one for that how the school site. But to do this, that would be proper tips, proper that stuff. Last but not least, and so PCR has been a very famous method that gained attention from us since masa era. Even orang yang ada tak cerdik dia ada. PCR, polymerase chain reaction, the most frequent phenotyping technique. robbers from screening for its fast, specific, rapid. It has provided the opportunity to provide science and things. DNA are very specific. The whole blood collection is completely true. So how do we do a PCR for this? Right, the 10% blood volume. Yeah, yes. First of that, DNA extraction. So DNA extraction that is blood. There are many DNA extraction hit targeting. For example, I use protein oil in what you do is you take the protein, take how many ml of blood in one, like the whole blood, blood, take put the lysis solution, remove all the antigens, and get pure. Then based on the primer that you need, whatever you target, then you can do either a simple PCR, you can do a nested PCR, nested you target the gene together. The method is actually you are targeting a small gene into a big gene. So primer, multiplex PCR. So it depends on whatever target you want. So we have different right? For example, 18S RNA is one primer that is quite conserved, specific. This other. What are the other techniques? Data picture is sheet that you can use. So, consider the level of current. Multiplex next step. That assay has been developed. These are all molecular based detection. It's developed based few years ago, quite famous, not to say too. But it has not been very oftenly used for testing. This is a very good assay. You can do it at field sample, but not tried yet. The only thing drawback about live assay is you need to synthesize the primer around 6, 8, many primers using some gadgets. So this has not been used as a diagnostic, official diagnostic method. But some disease they are still trying to use that. So now let's compare between microscopy, RGG, and PCR. See, you have microbiological interpretation, antigen, antibody binding. These are the main difference between microscopy, RGG, and this one you must be very clear. The golden standard method is still target diagnostic. Microscopy, you can detect all stages of the guardian. As long as you know how to get the slides, and it RTDs get all the proteins. specific to malaria, and of course for PCR, locality, whichever region that. In terms of sensitivity, so what's the difference sensitivity and specificity? Sensitivity indicates uh, as much as little a parasite can be detected. Berapa yang paling kecil kuantiti parasite boleh detected. That is called sensitivity. Specificity bermaksud adakah yang spesifik hanya ke virus sensitivity is microscopy 5%, RTD 50 RTD actually is not sensitive. You need quite a high amount of parasite. PCR is very sensitive, below 5 parasite. What about specificity? Microscopy, yes. They are actually specific. They are good in reading the dogs and differentiate. Only knowledge side, you need some a good idea how to differentiate human malaria and monkey malaria. Specificity is modern. As I showed, some kids are only able specific for falciparum. Yang lain kita actually ada corfan malaria. So they akan highlight another string. They overlay again. PCR, the hand, and that's the time consuming up to 60 minutes. Very quick. Tarik, tail nicely, stain it and observe. RTD very fast. This will take time because it takes time to extract the DNA. Interpretation, quantitative, quantitative and qualitative. Can be both quantity and quality. Here's where advantage to pause and keep the slide for a long time. RTD is very fast practical. You can what mana-mana. Kid take the blood sample, even cleaning the dalaman you can just. This requiring response the DNA needle, but this advantage is need special equipment and training technician. Equipment I wouldn't say it's so difficult, it's just a mic. But to get a good malaria screening, it takes years to train. Even me at times I'll ask confirmation with other malariologists from other institutes. The advantage for RTD cannot be used for drug monitoring and RTD can be very. You can just say yes, but whether the patient has quantity, berapa seriously, PCR, your machine, you need the DNA extraction kit, and you must be a very clean DNA. Molecular methods to get contamination is very difficult to troubleshoot. You must have someone really have patience, perseverance, and enough because the volume that your pipetting is very minute. 1 micrometer, 1.5. So, and you need to focus in that many notes, especially when you have many pockets. Yeah. Okay, so in summary, the slide. Microscopy is still the golden slide here. Things microscopy is still RTDs are effective, but they have like cause death. ELISA tests are not good for flow cyto can be used for specific but requires well-trained techniques. PCR-based tests. So flow cyto is not even included here. It's under flow, it's under like antibody detection, but it's really expensive. Not that and it's not that fast. Get flow cyto, you must do the proper staging. For research, yes, you can use. For diagnostic, it can, but it will take some time to prepare the test. PCR-based test should be the choice for testing whenever you can get information of parasite growth, species, resistance, but you still need the proper training. So you have various real-time PCR, multi-implex PCR, method, different interpretation. So when you use multiplex, so real-time we use multiplication and to see the level of parasitism. If the CT value is very fast, get the CT value quite quickly, it shows that the patient is high infection. Multiplex you use to detect various parasites at once. There are various primers. So the primer design should be very meticulous, accurate, can't mix up. And that's the PCR. Right? Nested is actually you have to amplify one single gene. So you amplify the big part of the gene. Okay. So last but not least, thank you students. Thank you so much. Thank you UNISA. So we shall open to question and answer session. Is there any burning question from you students? Is there any questions from you? Anything about malaria? I will cover here.
Okay. Thank you for the extraordinary. Thank you for the extraordinary presentation for materi by Dr. Mencerdaskan, PhD. Silakan untuk teman-teman yang ingin bertanya, kami persilahkan. Bisa unmute atau tulis pertanyaannya pada kolom komentar.
So everyone understood very well, no questions. I know, I understand this is a Friday. So what our conclude is malaria still is because golden standard method is still microscopy. It takes a long time to diagnose. You need a proper technicians, well-trained technicians and have and all the other factors that are is that PCR and so on, they have been used. Like for example, last time they used real-time PCR here, but flow cyto all is still some they use it. By answer somebody on students. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Sebelumnya perkenalkan nama saya Laisa. Saya izin bertanya pertanyaannya, bagaimana cara kita mencegah penyakit malaria bila kita harus tinggal dan bertugas di daerah endemik malaria? Terima kasih.
Pencegahannya, first of that, jikalau anda sudah mengetahui tempat itu adalah epidemik malaria, okay, and anda ditugaskan di sana, first, always wear a protective clothes, pakaiannya, clothes, pakaian, clothing lah. Use mosquito repellent. Nyamuk anopheles, they don't like the chemical smell from mosquito repellent. Mosquito repellent pun the spray untuk mencegah nyamuk. Semasa anda bertugas selalu ada sebotol spray. Do fogging. Pastikan authority yang tahu anda bekerja sana buat fogging untuk hilangkan ni. Tidur, masa tidur, mosquito net. Sekarang kita ada mosquito net yang dibuat menggunakan kaedah bioteknologi, sudah ada chemical untuk hindarkan nyamuk. Mosquito net. So kalau boleh jangan keluar lepas waktu saja. Nyamuk anopheles suka menggigit sajalah. Okay, that's all for the prevention. Kalau ubat itu ada.
Thank you.
Regarding ubat, I'm not sure about Indonesia. In Malaysia, ubat pinon tidak dibenarkan ambil. Kita tidak boleh beli dari farmasi. Hanya hospital kerajaan. Kalau anda ada penyakit malaria, kita boleh makan. But saya tahu setengah negara ubat pinon itu dijual di kedai, like Bersama and so on. Here we do that. Gejala resistant. We do sell pinon. Ada.
Pertanyaan berikutnya yaitu resiko komplikasi apa saja yang bisa terjadi dari penderita malaria. Pertanyaan berikutnya yaitu kapan waktu pengambilan sampel yang baik untuk diagnosis malaria? Mohon maaf Ibu, apakah itu apa di mute atau gimana ya Ibu?
Oh, sorry, saya di mute. Okay, jawapan dia untuk tu, sorry, I didn't feel I was muted. No, speaking. Okay, jawapan dia ialah malaria kita boleh ambil bila-bila masa sahaja untuk sampelan. Kita tidak mengikut waktunya. Bukan macam peliharaan. Take at all times. Kalau seorang sakit disyaki ada penyakit malaria, kita boleh ambil sampel dan
Mungkin untuk teman-teman yang lain ada yang ingin ditanyakan, boleh silahkan unmute atau diketik di kolom komentar pertanyaannya. Baik, sepertinya tidak ada lagi yang ingin ditanyakan, maka saya akan akhiri. Terima kasih kepada pemateri atas materi yang sudah disampaikan. Semoga bermanfaat buat kita semua untuk kedepannya. Dan terima kasih juga untuk teman-teman yang sudah bertanya. Saya kembalikan ke MC.
Okay, thank you.
thank you. Thank you, moderator.
The next event is the symbolic handover of the research person certificate.
Very good. Thank you. Third student.
Eh, untuk teman-teman bisa kameranya bisa diaktifin dulu. Sekarang kita mau sesi dokumentasi terlebih dahulu. Oke, senyum. 1, 2, 3. Gak ada yang Oke, terima kasih.
Okay, thank you students. Have a good day. Bye. See you again. Jumpa lagi.